So this afternoon, we're joined by Dr. Robert Nagourney and Dr. Paul Kent. Dr. Nagourney has been working with FibroFighters now for the past 4 years on a functional profiling, and it's been extremely successful. But we're now at a stage where we're moving into the metabolomics, and we have a great new exciting program that Dr. Nagourney would like to speak a little bit about. So Dr. Nagourney, why don't you let everybody know what our next project is?
Well, thanks, Tom. First of all, Paul, it's a pleasure to have you visit.
Thank you so much.
We just met a few weeks ago at the American Association for Cancer Research meetings in Chicago, and here we were able to bring you out to visit. So yes, Tom, as you mentioned, we've been working together for the last several years studying basically human phenotype, the study of cancer biology at the level of cellular response to injury. And as we've crafted drug combinations, and Paul has been so instrumental in identifying the drugs and combinations tested, we begin to realize we're seeing signals that may reflect metabolic changes in these cancers. And if that's the case, then the question becomes, could we examine the metabolic basis of this? And that's what we're looking to launch now, which is our metabolomic analysis. And we're happy to be able to give you a tour of the lab and be interested in your comments, because I think metabolism may offer a new insight into this disease.
Can you explain to our audience what you mean by metabolomics?
Study of human metabolism. Life is energy.
Yes.
And in the human body, energy is metabolism. So when we study, just as genomics is the study of genes, metabolomics is the study of metabolism. So what we're using is a technique to measure metabolites. These are the intermediates in the energy production in the body that enable us to know what nutrients are being used and what byproducts are being produced. And we can study that both in the blood, plasma, as well as in the tissue culture environment. And that's kind of what we're looking at now.
RNA, and we started to learn in cancer that's the starting point, but it turns out that just going after the DNA or the RNA or even the proteins that come next still wasn't getting us to the promised land. How can we actually reliably kill cancer? Now this is more complex, but also so much closer to reality. Now we're going after, how does that cancer cell stay alive?
Exactly. Yeah, that's really exactly the point. What our laboratory measures is programmed cell death.
Yeah.
So as we were studying these fibrolamellar cases, we were looking at cancer cells injuries leading to cell death. And then as we dove deeper into that, we realized that the death that was occurring is a metabolic death. The cells were losing energy. So we said, well, if we could get down to the nitty-gritty of what they're using to make energy, and maybe even begin to use drugs that would block that process, perhaps we would have better treatments for these kids.
Yeah, the cancer cells have so many ways to defeat us and so many ways to stay alive. And trying to understand, why do they live when normal cells would have died? And turns out it's their own internal breathing apparatus, if you will, and how they eat and how they feed and how they grow, and they manage to stay alive when normal cells wouldn't.
That's Cancer cells can exist under conditions that normal cells could never.
Yeah.
They, for example, can exist at 1/10 the normal oxygen tension.
Right.
So in order to do that, they have to develop tricks, and all those tricks may allow them to stay alive under the right conditions. We think it may also provide vulnerabilities. So in the future, perhaps we can trick these cancers into dying without necessarily poisoning them.
That's amazing.
So how many patients have we had this metabolomic study performed on thus far, and how many do you plan to use in your study?
Well, we've only had the opportunity to study about 2 or 3 patients with this plasma metabolomic platform. It is not a difficult study to conduct at our level, but we need to get the plasma. So what we've been talking about today is the mechanics of getting people to send us plasma, and then we would run it through these metabolism studies, and we might also, where possible, test the metabolism of the tissues in culture, so that we could look at the microenvironmental metabolism. And putting those together, I hope, might give us insights for treatments, for opportunities for therapies, maybe even less toxic treatments.
What is plasma? How's that different than blood?
Well, as you know, the blood is made up of red cells, white cells, platelets, and quite a lot of liquid. And the liquid in the blood is the plasma or serum, which is the kind of yellowish liquid when the red cells and all the other cells respond down. So in a centrifuge, if you spin down the cells, you get a pellet, you get a bottom of red, thick blood and liquid, and that's the plasma.
I see. So we need our patients and their doctors to just to centrifuge
Right.
a blood sample as the first step for
Right.
you to study it, right?
Right.
Yeah.
And then ship it overnight on
Great.
ice, and we'll get them, stockpile them, and then run them as batches.
That's wonderful.
And we're hoping if we could get between 10 and 20 of these cases studied, I think we'd have some brilliant insights into this disease. I think personally what we're seeing is that there are very significant metabolic signatures. This disease has a metabolic signature, and I would love to try to exploit that as a therapy.
I know that our group will probably be asking a question. Can we get some type of an answer specifically like we do from the functional profiling? Is there an answer that's a part of the metabolomics? So will they get a report?
We are very happy to provide a report. We report currently on 42 individual metabolites and almost 120 additional studies that we conduct. The report that we would provide would be amino acids, chemicals in the blood called biogenic amines, hexoses, which are sugars, and then fats called acylcarnitines. And these constitute measures of how the cell is making and using energy, and where there are excesses or deficiencies, perhaps we could begin to craft interventions that could supplement people with dietary changes or micronutritional changes, and maybe some drugs that would lead to metabolic injury to the cancer cells. That would be hoped.
Wonderful. Thank you so much.
Tell me about glutamine and fibrolamellar.
Well, glutamine, as you know, Paul, is
Yeah.
the most abundant amino acid in the human body. Glutamine itself is involved in an enormous number of reactions. So among the reactions is glutamine becoming glutamate, glutamate becoming alpha-ketoglutarate, and alpha-ketoglutarate going directly into the mitochondria and taking the place of sugar.
Yeah. Okay.
So when a cancer cell is deprived of sugar and is looking for a nutrient, it can go to glutamine. And if it doesn't get the glutamine it needs, or if it doesn't get enough glutamine, then it will go to lipids, and then it starts chewing up fats. So these are all the interconnecting pathways of energy production, and glutamine is fundamental to that. So one of the avenues of therapy now is to inhibit glutamine.
Mhm.
And this compound DON that we're currently going to be studying, and the glutaminase inhibitors will give us some leads on how important glutamine is to this disease. Now, not every cancer relies on glutamine. For example, we know that the glandular cancers, breast cancer and prostate cancer, are very glutaminolytic. We know that from our data. I don't know enough about fibrolamellar to know how glutaminolytic or how much they rely on glutamine metabolism, but we'll know. We'll find out soon.
Wonderful.
That's wonderful.
Well, thank you very much for answering these questions and for your time this afternoon, Dr. Nagourney. We really value it, and we value you, and I know our community does. So thank you today very much, and thank you, Dr. Kent, for joining us. We sure appreciate it.
Our pleasure.